Hi, my name is Clay Miner, and I'm going to be doing the MC1R protein mutation where we see arginine 67 get replaced with cysteine. So first off, we're going to be looking at the protein location and disease. We're going to see that the MC1R protein is located in the plasma membrane of melanocytes, and these melanocytes are responsible for the production of pigmentation, and this often occurs through the interactions they experience with hormones like the melanocyte stimulating hormone, which is directly related to the production of melanin. And melanin is the pigment that is responsible for skin, hair, and eye color, and these cells are often found in the epidermis of the outer layer of the skin, which is where we commonly see it and where we're looking at today. The MC1R protein is found across a variety of vertebrates, commonly in humans and mammals, but obviously it's in other organisms that are experiencing darkening or stuff like that with UV exposure. The condition associated with the MC1R that we're looking at, protein that we're looking at, is the replacement of arginine 67 with cysteine, and we'll go into detail on that, on why that switching causes the fur of a mouse to lighten from the wild type being a very dark brown to it being a lighter brown. And that has to do with the disruption of the pathway of producing melanin. So looking at the normal protein function, MC1R falls into the receptor category, and it's categorized as a receptor because it binds to ligands and triggers signaling pathways in response to cellular processes, like the production of pigmentation. MC1R is located in melanocytes, as mentioned earlier, and kind of the main function we look at for it is it binds to the melanocyte stimulating hormone, and this hormone then activates the receptors and triggers a signaling pathway that involves the production of cyclic AMP. This activation then leads to the production of protein kinase A, and then the synthesis of eumelanin. And eumelanin is the subset of melanin that produces the dark pigmentation that protects from UV rays. And this protection is crucial because it makes the top layer of cells darker, which prevents the cells from underneath and others from experiencing DNA damage from prolonged exposure from harmful UV rays. And then the mutation, which we'll see with this red amino acid right there. This is originally arginine, so if it was the same, this would have been blue. Obviously, we'll get in a little more detail in the model later, but that was switched to cysteine, and we'll discuss shortly how the different qualities of this impact the actual function of the protein. But the mutation of MC1R affects the receptor's ability to properly interact with hormones such as the melanostimulating hormone. And in such cases, the mutated version of MC1R can produce less activation with binding and signaling. This would typically lead to less melanin production, which is why we would see the lightening of the fur in the mice. And this would be, obviously, it would be drastically less production of melanin than you would see in the wild type, hence why the mouse did become lighter. Now looking more specifically into the amino acids that are the normal amino acids, so the arginine amino acid. Arginine is typically hydrophilic due to the positively charged guanine groups, causing a high polarity which increases the amino acid's interaction with water. And because of that, we see it having a positive charge at pH 7. It's also one of the larger amino acids, right around 174 Daltons. And one kind of the bonding qualities and other qualities of arginine is that its positive charge allows for it to interact with negatively charged amino acids like glutamic acid and aspartic acid. And these form salt bridges, and these salt bridges help stabilize proteins as seen in like the non-mutated MC1R protein. So the stabilization is what allows for its binding with the melanocyte stimulating hormone. Additionally, the guanine groups can act as hydrogen bond donors, which also aids in the stability, and that's because of the multiple amine groups that are attached to that. And this creates a strong, obviously this hydrogen bond creates a strong bond with other polar amino acids and kind of forms a more stable backbone of the protein. Now, looking at the mutated amino acid, which would be our cysteine, we see that cysteine is hydrophobic and not hydrophilic, opposite of the arginine. It is hydrophobic despite having a weakly polar sulfur group at the end. This doesn't produce strong hydrogen bonding and creates weakly hydrophobic environments, which would lead to it being neutral at a pH of 7. It's moderately sized, sitting at around 121 kilodaltons, which is about 53 less than we see with, or not kilodaltons, daltons, which is about 53 less than we see in arginine. So it's a smaller amino acid, and since we see this mutation occur in the side chain of the amino acid, it shifts from hydrophilic with a positive charge to a neutral with hydrophobic, a neutral charge with hydrophobic. There is significantly less interaction with other side chains, so you see the structure kind of break down, you see it not be as stable, and you see it often interact with those stimulating hormones less. It also allows for the formation of disulfide bonds with other cysteines in the amino acid chain of the primary structure. These can reduce polarity and are more hydrophobic compared to the other interactions you would have seen with the arginine, causing it for it to interact less with the stimulating hormones. So kind of the impact we see of the mutation is that the protein, specifically in the protein itself, is the change of arginine to cysteine results. We see this shift in coloration because this shift from arginine to cysteine is weakly polar and hydrophobic interactions that the cysteine produces, and these properties cause weak stability in the protein and disruption of receptor signaling. And furthermore, the neutrality of cysteine causes for poor electrostatic interactions that alter protein folding and stability in a negative manner. Another impact would be that cysteine replacement in that side chain can now interact with other cysteines and create those disulfide bonds that were mentioned earlier, which are very strong and hard to break once they do form. Specifically in the melanocytes and the cells where we find these proteins, typically the MC1R protein is responsible for binding melanocortins, or like the alpha melanostimulating hormones, and this process produces the dark pigmentation we see in the skin called eumelanin, as we mentioned earlier, which does provoke that UV resistance. And when cysteine replaces that, we see less eumelanin being produced, and you see more likely UV damage to DNA, which forces unfolding of DNA in other cells. And this is kind of a loss of function, and we notice that the mouse's fur does get lighter, and that's because the cells are producing less eumelanin. And then overall, looking at the organism, it's a loss of function mutation that occurs. It promotes pheomelanin, which is the counterpart, which is kind of the counterpart of eumelanin. It's what results in the lightening of the hair. This is seen in the mice examples, as the organism with the mutation have a light brown complexion as compared to the wild types have a very dark, almost like dark brown colored hair. This lack of pigmentation in the skin and fur can progress past just light hair in the organism, and you can see all-out albinoism, which is where there's no production of eumelanin. These organisms have a much higher risk of harmful UV damage. Luckily, the MC1R protein mutation we're looking at doesn't always result in albinoism, it just hinders the promotion or the production of eumelanin through the stimulating hormone, which is why we get lighter hair compared to a full-out albinoism. So this is often seen in it is less detrimental to organisms that live in lower UV environments, which are usually further from the equator, because this lack of pigmentation helps with their vitamin D consumption since they aren't experiencing so much direct sunlight. So the lightening of their fur allows for them to receive more vitamin D, which is necessary for their function. And then looking at the model here, this is the human MC1R locus. It encodes a 7 transmembrane, which are the toilet paper roll looking things. These transmembrane proteins are highly polymorphic. And the mature MC1R protein is a G protein coupled receptor that spans the membrane 7 times. That's why you see 7 of these. You also see extracellular. It's kind of the blue in the background represents the plasma membrane, and you see that there is extracellular loops and intracellular loops. The extracellular and transmembrane domains engage the MC1R ligands, while the intracellular and transmembrane domains regulate the adenylyl cyclase interactions and signaling. As mentioned, it has the characteristics of the G protein coupled receptors, the 7 transmembrane helicases. There's 3 extracellular loops. There is an extracellular amino N-terminus, which is seen up here, and then a intracellular C-terminus domain down here. And then the ligand binding pockets are seen in the extracellular loops up here. So this is the representation of the protein. Here is seen in the red. This is where the spot of the shift in amino acids goes from arginine to cysteine, and you can see it's in the intercellular loop where it would be reacting more with that melanocyte stimulating hormone. Looking at some of the model limitations, as for simplicity's sakes, all of these amino acids, which are the primary structure of the protein, there would originally be 317 of these. Obviously, that was not feasible. So this would not accurately represent the ability of the different amino acids to interact with each other and things like that. But the loops represent the primary structure of the amino acid. And this is often contains several important binding sites, specifically for the ligands as the alpha melanocyte stimulating hormone. Looking at the loop regions specifically, this is kind of where you start to see local folding proteins. This is the secondary structures, and you see these in the loop regions. And then in the 7 transmembrane helicases are the tertiary structures, and quaternary structures aren't necessarily needed for the actual MC1R protein. It mostly is just tertiary. But these transmembranes allow for the ligand binding pockets to form. And as you can see, 3 extracellular, 3 intracellular. The extracellular is important for ligand binding, and the intracellular interacts with the G proteins to trigger intercellular signaling, to trigger intercellular signaling. But looking at the limitations of the model, as mentioned earlier, there's not 317 amino acids in this primary structure. That would mostly, the drastic understatement would greatly impact the ability of the extracellular loops to interact with, or to the ligand binding to occur, since it would be inaccurately represented by the amount of interactions that would actually be going on with the folding and the stability of the structure. Another one is the amino acid primary structures actually pass through these transmembrane areas. And these aid in the structure of the protein and ligand binding and signal transduction. And the seven transmembrane primarily consist of hydrophobic acids in order to create a more stable protein. Another one is that all of these altoids that have been painted would be more like, there would be different colors because there's a array of amino acids within the protein, and for simplicity's sake, these are all painted similarly to highlight the mutation, but these would be painted different colors, or these would be representative of different amino acids with obviously repeats throughout. But those are the main limitations of this model.